Hi and Assalamualaikum. My name is Harissa Shazana binti Muhammad Nur Azmi. My metric number is 194611 and I will be presenting my journal club presentation entitled Degradation of Polyester Polyurethane by an Indigenously Developed Consortium of Pseudomonas and Bacillus species isolated from soil. So this is the screenshot of my journal. It was published on November 4, 2016. So without further ado, let's move on to the topic introduction. So what is polyester polyurethane? It is a synthetic polymer that is used as raw material in different major industries such as construction, transportation, furniture, packaging, and many more. It is made up of three components: polyisocyanate, a polyol, and a chain expander by condensation reaction. The production of polyester has been 10 million metric tons annually, hence has raised concerns due to its high resistance to degradation. Although it is susceptible to both bacterial and fungal attack, it is also known for its strength, abrasion resistance, and chemical resistance. It is divided into two segments: hard segment that contributes to hardness and soft segment that contributes to elasticity. So there are two bacteria used in this study to construct a consortium. The first one is Bacillus subtilis, which is a gram-positive bacteria, strain MZ75. It can be found in soil and gastrointestinal tract of ruminants and humans. Next is Pseudomonas aeruginosa, a gram-negative rod-shaped bacteria, strain MZE85, also can be found in soil. So these two have the same ability, which is to degrade polymer. This is due to restriction to its soft segment in individual capacity without evidence of attack on hard segment. So the problem statement is, although polyester polyurethane is considered as biodegradable plastic, the microbial degradation process can take up a longer time compared to other polymer due to their high resistance to microbial degradation. So the aim of the study is to determine the degradation efficiency of bacterial consortium, Bacillus subtilis, and Pseudomonas aeruginosa against polyester polyurethane in comparison to the individual strains. So moving on to the methodology. First of all is the preparation of polyester polyurethane. Okay, so polyester PU films were prepared by solvent casting, which took about 3 days to form thin, transparent polyester PU films of uniform 0.2 mm in thickness. Next is preparation of consortium. So this can be done by inoculating a loopful of each strain into two 500 ml Erlenmeyer flasks containing 100 ml of lithium broth separately and incubated overnight at 37 Celsius. Next is polyurethane degradation assays. So this is divided to three parts. First is the measurement of weight loss, to measure the weight loss of the PU film every 3 days interval. Second is scanning electron microscopy to analyze the changes in surface morphology of the PU films. And the third one is carbon dioxide evolution test to utilize the formation of CO2 as a result of oxidation of carbon by microorganisms. Next is esterase activity assay to determine the specific enzyme activity. And lastly is GCMS, which stands for gas chromatography mass spectrometry analysis of degradation products to detect 1,4-butanediol and epidic acid. So moving on to results and discussion. First is the measurement of weight loss. The weight loss was due to the breakdown of long PU chains into low molecular weight fragments. So as you can see here below, this is the plot of the consortium, shows a significant drop of weight. So hence, it shows that it is more efficient as compared to individual strains in degrading PU film. Next is scanning electron microscopy. So A is the control, which means no change in surface morphology. B, C, and D is the same PU film with increasing magnification. So as you can see here, there are clear pits and holes and also biomass attached. Biomass here referred to the bacterial cells. So the adherence of bacterial cells to polyester PU film pieces, as depicted in this figure, indicated the tendency of bacteria towards polyester PU in order to utilize it as carbon and energy source. So moving on to carbon dioxide evolution. It is also known as Stern test. During this test, the microbial degradation of polymeric material can be evaluated by entrapping carbon dioxide evolution as a result of mineralization. In this study, the quantity of CO2 released in the test vessel containing polyester PU as the only source of carbon was significantly higher than the control vessel without polyester PU films. So here shows that the high amount of CO2, which is 8.16 gram liter, was recovered after the quantification. Next is esterase enzyme activity. Esterases Esterases were produced by the bacteria upon utilization of the PU films. So esterases work by attacking and damaging the surface of the PU film through hydrolytic degradation, which resulted in the formation of holes and pits. So the higher the concentration of esterase detected means there are more formation of holes and pits of the PU film. So the synergistic increase in enzyme activity were attributed to co-metabolic interaction between the members of consortium, resulting in better substrate assimilation, higher productivity, more flexibility in changing conditions, and increased resistance to contamination. So here shows the esterase activity consortium, which rise faster than that of the same organisms used in pure culture. Next is gas chromatograms analysis. So as mentioned earlier, it is to detect the polyester PU degradation products, which are 1,4-butanediol and adipic acid. So below shows the GC chromatograms of the consortium, which shows high intensity value of both 1,4-butanediol and adipic acid compared to these two. Okay, so it can be concluded that the rate of degradation was enhanced when Bacillus subtilis and Pseudomonas aeruginosa were used in the form of a consortium. The high amount of esterase produced by co-culture consortium resulted into effective degradation of soft and hard segments. So these are the list of references. I hope you could understand my presentation upon this topic, as it is a very simple study. So I hope you enjoy it, and thank you for watching.